Well, I hope I convinced you in this last video, and if you haven't watched the short video on day versus night for plants, I encourage you to do so. But I hope I've convinced you that it's really important that chloroplasts only be active in doing their biochemistry during the day, and at night for plants, mitochondria is going to take over. And so the question sort of becomes, there's certain aspects of chloroplast biochemistry that you can just control by there not being light available. But then there's certain enzymes that we need to make sure are off to avoid a futile cycle. So how do we do that? Well, there's lots of enzymes that are involved in particularly the Calvin cycle. That's the piece that we need to make sure that we control well. So this whole idea of the light reactions and electron transport, that's not going to happen if the sun's not out, but we have to make sure that the Calvin cycle is inhibited. So how do we determine how we find out how we inhibit certain metabolic pathways is we look for flux control points. So hopefully that's not something that's unfamiliar to you. But when we think about identifying flux control points, what we need to do is look for enzymes that are operating far equilibrium. Hopefully you remember too, this is not a set of data that we're going to be looking at because delta G naught prime represents standard state conditions, and that's not really where we operate generally for biochemical pathways. We're not at 25 degrees C, we're not at 1 molar concentrations. So we are going to be looking at the column that is delta G that is under the conditions that we're used to be thinking about, sort of those biological conditions. And if we look here, hopefully we can easily identify, let me zoom in here because it might be a little bit small there. We're going to see 3 reactions that have large negative delta Gs, okay? One of those is a guy that we've talked about already. Here's Rubisco. And again, I want you to remember that Rubisco is the magic money enzyme that's in the Calvin cycle that does our carbon fixation chemistry. This is the enzyme that takes carbon dioxide and attaches it onto a 5-carbon carbohydrate and ultimately begins that process of carbohydrate synthesis. So key enzyme that we have here. These other ones, and you can see there's a laundry list of enzymes that are part of the Calvin cycle. Just like there was a laundry list of enzymes that we had for the pentose phosphate pathway, we didn't learn about every single one of those enzymes. If you go and look at some of the names that we have here for some of these enzymes, they'll look familiar to enzymes that we see in the pentose phosphate pathway. But one of the things we're going to note is that there are 2 enzymes here that operate far equilibrium, and I want you to know what these are. We can use sort of the abbreviations. FBPase, that's an enzyme we know already, and then SBPase. Okay, so 2 phosphatases that need to be controlled to make sure that the Calvin cycle is not operating when it shouldn't. So I want to take a minute here and highlight how we control these different enzymes. So we're going to get to these guys in a minute. These are operated or are controlled via redox cascades. Okay, so we're going to talk about what a redox cascade is, and we'll see how that is used to sort of control activity of these 2 enzymes. Rubisco is a little bit different, okay? Rubisco, we can think about there sort of being like a 3-step check to make sure that Rubisco, this is such an important enzyme, we really want to make sure that if it's time for Rubisco to be active, that it is. So here are the 3 ways that we can control activity of Rubisco. So Rubisco is a pH controlled enzyme. Okay, so again, we only want Rubisco to be operating when it's light out. Rubisco lives in the stroma. So let's make sure to highlight these pieces. So Rubisco lives in the stroma, and we only want to be active when it's light out. Okay, let's move these guys. I guess we'll leave them there. Okay, Rubisco lives in the stroma, and we only want it to be active when it's light out. So let's think about what the pH of the stroma is going to be like when it's light out. When it's light out, we're doing lots of electron transport, which means we're doing lots of proton translocation to generate a proton gradient, which means the concentration of protons is going to be low in the stroma, which means pH is going to be high. So when the pH is high, we are going to be activating Rubisco. Okay, so it has a pH optimum around 8. You don't need to remember that number, but that helps remember that when pH is high, we want Rubisco to be active because we're coupling together those light reactions with Calvin cycle chemistry. Okay, pH is one way that we control. Another way that we control is magnesium concentration. And when magnesium concentration is high, we are going to be activating Rubisco. Here's the logic for this. When we are doing a lot of electron transport chemistry, pH again is going to be high because we're moving those protons out. But remember, we are also then moving those positive charges out. That creates a positive charge, or could create a positive charge on the outside of in our inner membrane compartment. But that doesn't happen in chloroplasts because of the inability to build up a charge gradient. And the reason is magnesium is able to move then into the stroma. So magnesium is going to move into the stroma as a way to balance the attempted building up of a charge gradient. So as protons go out, those positive charges go out, magnesium as a positive charge is going to come in. So the fact that magnesium activates Rubisco is sort of that connection between, hey, that's good, we're going to have high levels of magnesium in the stroma when it's light out. The last piece here is a transition state analog called C1AP or CA1P. And if you don't remember this, don't worry about it. Just remember that this is a transition state analog. We learned about transition state analogs when we did our enzyme kinetics learning unit. Transition state analogs bind very tightly to enzymes because they resemble the transition state of substrate going to product. So transition state analogs are often used for drug design and so forth, but plants synthesize a transition state analog for Rubisco at night. So this transition state analog is synthesized at night. So sort of a three-step process here that we have to have a high pH, high magnesium concentration, and the absence of this transition state analog to ensure that Rubisco is active. Flip side, when it's dark out, we are going to have an acidic pH in the stroma, low pH, because we're not translocating protons, we're not going to be moving magnesium, so we're going to have a low pH in the stroma, and plants are going to synthesize this transition state analog just to make sure in case there's any pH or magnesium levels that would activate Rubisco, it's going to be quiet. It's so important to not have that futile cycle. So that's how we control Rubisco. So let's talk about this redox cascade piece and understand how we control activity of these two phosphatase enzymes. So this is a good opportunity to do a little bit of a review. So what I have on the left here is a phosphorylation cascade. So we're just going to run through this phosphorylation cascade. As a way to review how cascades work. Okay, so we've seen this before. Let's zoom in here and sort of talk about what's happening. Okay, we're going to have a stimulatory external signal. So we're ultimately talking about the activation of adenylate cyclase. So an example of a stimulatory signal might be glucagon. So let's walk through sort of what happens here. And so this is going to be an opportunity to review sort of the main phosphorylation cascade that we've learned about in biochemistry here. So we're going to have glucagon. Glucagon is going to activate adenylate cyclase. That is going to increase the concentration of cyclic AMP. Okay? Cyclic AMP is going to activate pKa. Remember, pKa was that R2C2 enzyme. Okay, and if as I'm saying all of this, you're like, this is totally Greek to me, you want to make sure you go back and review that because this is kind of one of those things that's important to sort of think about long term. PK is going to activate phosphorylase kinase. So phosphorylase kinase. Okay, that is the enzyme that has the alpha, beta, gamma, and delta domains. Remember those alpha and beta domains, these are the ones that want to be phosphorylated. We've got our gamma domain is the one that wants to bind, our delta domain, that calmodulin domain, that's the one that wants to bind calcium. And ultimately, this gamma domain, that is our kinase domain. Okay, that kinase domain is going to activate phosphorylase. Phosphorylase is the enzyme that we're going to be phosphorylating on that serine 14. And ultimately, when that serine 14 is phosphorylated, it's going to be locked in with those arginine residues. And so that N-terminus is going to not be buried. It's going to be out. We are going to break down glycogen. And at the end of the day, this is going to increase the concentration of glucose. Point being, cascades allow us to connect an upstream signal with a downstream solution. Okay, so this was our phosphorylation cascade. Kind of running through it here, if this is glucagon that we have here, we really didn't talk about these G protein signaling pathways here. But ultimately, adenylate cyclase is going to be activated. We're going to have pKa here, that's our R2C2 enzyme, be activated. And here, this very generically talks about phosphorylating other proteins and having a downstream cellular response. Okay, so let's talk about what the options are for this redox cascade. Okay, so maybe you remember when we talked about mitochondrial electron transport, we talked about how we controlled flux through mitochondrial electron transport. It's easy for chloroplast electron transport, just turn on or off the light, right? But for mitochondrial electron transport, ultimately, the redox state of cytochrome C is going to determine flux through mitochondrial electron transport, whether or not cytochrome C has those electrons. Okay, so at the end of the day, looking at a certain molecule and whether or not it has electrons can determine whether or not a pathway is activated or inhibited. Okay, so let's think about our chloroplast electron transport and thinking about all of the carriers along the way. What is going to be the important piece here is what ferredoxin does with its electrons. Okay, so thinking about ferredoxin, that is almost at the end of our pathway, but ferredoxin has 3 possible options for what it can do with its electrons. So let's talk about ferredoxin and what it can do with its electrons. So just as a way, I'm going to zoom in. We're going to look at this picture that's over here. Okay, so this basically shows us sort of electrons going through different carriers in chloroplast electron transport. Okay, so if we've got light, if there's light out, okay, we are going to be doing the electron transport chain in chloroplasts. So what we're going to see here is we're going to see things cycle between oxidized and reduced states. When they don't have electrons, they're oxidized. When they do have electrons, they're reduced. So sort of look at this pathway and this flow of electrons. So again, starting and looking at electrons from photosystem 1. Remember, this is not the start of the electron transport chain. This is the very end. So as photosystem 1 has electrons, photosystem 1 again is going to be activated when we have light shining at 700 nanometers, and we're going to excite those electrons. Okay, those electrons are going to be delivered to ferredoxin. So here, if we follow the blue here, I'm going to highlight this in blue here a little bit darker because it's really hard to see that this is blue. This is really following the electron path. Those electrons go from the end of photosystem 1 and they go on to ferredoxin. What that's going to allow this photosystem to do is go from a reduced state back to its oxidized state as it gives up its electron. Okay. Here's again the start. Here's ferredoxin. Okay, so ferredoxin right here. So I'm just going to label this red. Okay. we have a couple of options, 3 options to be exact, of where ferredoxin can take its electrons. I'm going to flip back just for a second here because I think this will be helpful. This was from a previous video, okay, but looking at the options for ferredoxin, this might be helpful. So ferredoxin is right here. Ferredoxin is right here. Here's ferredoxin, okay? Ferredoxin has the ability to take its electrons up over to FNR. Okay, so here's our options. This is what we're going to be talking about. We're talking about this redox cascade now. So we can go in a non-cyclic pathway and we can take these electrons over to FNR. It can say, hey, wait a minute, I want to go ride the ride again, and I'm going to give my electrons back to Q, allowing us to participate in the Q cycle again. That's going to increase ATP synthesis. Or they can deliver them to a partner in the redox cascade. That's what we're getting to right now. So let's go back to this right now. Okay, let's see, we're right here. So let's talk about these options for ferredoxin. We just kind of looked at them there, so let's write them down. We can deliver them to FNR. If we deliver them to FNR, ultimately that's going to be delivering electrons to NADP+, reducing it to make NADPH. Okay, and this is our non-cyclic pathway. Okay, we also have the ability to deliver those electrons back to Q. This is what we call the cyclic pathway. That's going to allow those to go on and then continue to build our proton gradient. So that's going to increase ATP synthesis. And the third option is ferredoxin could deliver them to our redox cascade. Okay, and so that's what we're talking about here with this blue line here. All right, so let's go back over here. So now we've got our electrons at ferredoxin. Okay, so here's what's happening is we literally, and we're going to call this an electron hot potato. So this is going to be an electron hot potato. Okay, so we're going to follow this blue line. So ferredoxin is going to deliver its electrons over here. You do not need to remember any of these names of these enzymes, but notice what's happening here. We have all of these guys who have thiol functional groups, probably from cysteine residues that are present in these proteins. Okay, once they go from their oxidized state here and they take on those electrons, they become the reduced state. So the oxidized state is going to be that disulfide link, and then the reduced state is back when there is free thiol. We do not need to remember the details of these players. Okay, we start out with this guy called ferredoxin thioredoxin reductase, and then we're going to deliver them to somebody else called thioredoxin. Here's the part that's important. The final sort of delivery is to one of these phosphatases. Okay, so here these phosphatases are either our FBPase or our SBPase. But look what happens when we are in the reduced state. That means we are active. So run through this logic check with me. We started out with a signal that it is light out. When it's light out, there's electron transport going on. When there's electron transport going on, ferredoxin can get electrons. When ferredoxin can get electrons, one of the places it can deliver electrons is ultimately to these phosphatase enzymes, SBPase and FBPase. When these guys get electrons and are in their reduced state, they are active. Remember, these are Calvin cycle enzymes. So when they're in the reduced state and they're active, that means the Calvin cycle is active. We are connecting then the ability for it to be light out with doing Calvin cycle chemistry. The flip side is true. If it is dark out, that means these enzymes are going to not be in the reduced state. They're going to be in their disulfide link state. And in being in that state, they're not going to be active and Calvin cycle chemistry cannot happen. Wow, I know that's a lot. So let's take a minute and try to summarize all of this. So I'm going to draw a little table here. And at the end of the day, we're going to talk about what happens at day versus nighttime and what's happening with our light reactions, what's happening with the Calvin cycle, okay? And then we're also going to bring in PFK, right? PFK, rate controlling enzyme of glycolysis. So we're asking the question, are we activating or inhibiting glycolysis? Okay, well, during the day, what should be happening with plants is that we should be activating the electron transport chain. We should be activating the synthesis then of ATP, NADPH, and then oxygen, which is also produced in the light reactions. And the cool thing is this is automatically controlled with the presence of sunlight. So we're going to call this auto control with, and I'm going to draw a sun here, because the sun is ultimately going to be determining whether electron transport can occur. And if electron transport can't occur, that means we're not going to be doing any of this stuff. And that's what we'd expect to happen at night, is that the light reactions should be off at night, but that's automatically controlled by the fact that there's not light. Let's talk about the Calvin cycle, right? We want the Calvin cycle to be active during the day, and we want there to be the activation of Rubisco, and we want there to be the activation of those SBPase and FBPase enzymes, because at the end of the day, we want to be activating the synthesis of carbohydrates, okay? And we've talked about how we can manage all of that, right? We are going to have the pH as a way to control Rubisco, magnesium concentration as a way to control Rubisco, and the fact that that transition state analog has not been synthesized will control Rubisco. We've got this connection with the fact that the electron transport chain is going on, right? That is ultimately going to be contributing to the redox cascade that's going to determine that FBPase and SBPase will be active. Okay, just highlighting and putting in here, we are going to have a high pH, high magnesium concentration. Those are ultimately going to be activating Rubisco. Now, because we have all of these mechanisms for generating ATP, we do not need to be breaking down our fuel reserves, so we should be inhibiting glycolysis by inhibiting PFK because it is not needed. All right, so let's finish out our nighttime, right? Nighttime, it's easy to control the light reactions because there's no light out, so there's not going to be any electron transport. But the key here is that we must find a way to inhibit the Calvin cycle. Okay, so how this happens, right? We need to inhibit Rubisco. So how we do that is we're not going to be doing electron transport. So let's just say electron transport is sort of automatically inhibited. Okay, when electron transport is automatically inhibited, that means we are going to have a low pH, we are going to have a low magnesium concentration, and we're also going to synthesize that transition state analog. All ways that we ensure that Rubisco is inhibited. We also need to make sure that we are inhibiting SBPase and FBPase. And we're going to do that by the fact that electron transport is being inhibited also means that we are going to be inhibiting that process because there's no electrons to put SBPase and FBPase into their reduced or active forms. And lastly, we must make sure at night that we are activating PFK. Because plants still need to have ATP and NADPH to sort of do their cellular biochemistry, what they need to do, though, is tap into those carbohydrate reserves that they made during the day as a way to make that through catabolic processes. And glycolysis is going to be the first step of that catabolism of those glucose-derived carbohydrates.